Hi everyone. This is a quick little lecture on chapter 4. I'm going to actually have two short lectures so that they're not too long, so that you guys can watch them and kind of see which one you want to see. So this first one is going to be an introduction to eukaryotic organisms, and I'll go through some of the external structures on eukaryotic cells all the way through to, I think, cell wall, and then I'll start my other mini lecture. So anyway, we're going to get into the history of eukaryotes first. The first of these types of cells appeared 2 to 3 billion years ago. Remember that bacteria, archaea, and also eukaryotic cells all evolved from one common ancestor. And the first primitive eukaryotic cells were most likely single-celled and independent. So you can think of them as probably a one-cell organism. And then over time, somehow these cells began to group together and form colonies. And as they grouped together and formed these colonies, some of the cells became specialized. And so perhaps some of the cells specialized in maybe breaking down nutrients. Others became specialized in protein synthesis. And what ended up happening was that these complex multicellular organisms evolved when these individual cells lost the ability to be independent and actually needed to be part of this intact colony. And it was those combined roles or combined functions that allowed this multicellular organism to survive. So the ones that we're going to look at in microbiology can range from unicellular to multicellular. The ones that are always unicellular are protozoa. There are fungi and algae that fall within the unicellular or multicellular, depending on either the type or what's going on with the growth of that organism. And then the helminths, which are the worms, are all multicellular, although they do have unicellular egg or larval forms, but generally speaking, they're multicellular. This is a great diagram from your book. It may not be completely clear to you in this picture because it looks a little bit small, but it's similar to that one we had of the prokaryotic cell last chapter, and this is kind of the same idea, giving you structures that would be found in all eukaryotes and then structures that might be found in some eukaryotes. So across the top, any of these purple labels are ones that you would find in all eukaryotic cells. And then the ones down here in pink are ones that you would find in only some eukaryotic cells. For example, a flagella. You wouldn't find that on all eukaryotic cells. So this is a great one kind of as a resource to compare to your prokaryotic cell. So I'd like to start with different structures that you would find on the external portion, just like we did with the bacteria, and move our way inwards. And the first two that we're going to talk about are two that would not necessarily be found on all eukaryotic cells. The first one would be the eukaryotic flagella. And this one is somewhat different from a bacterial flagella. First of all, it's much larger and it's much more complex. It's covered by the cell membrane, and it has these regularly spaced microtubules. Once again, though, its main function would obviously be motility. There are also eukaryotic cilia, and these are similar in structure, having those cylinders, those microtubules, like the flagella, but they're smaller and much more numerous, and these are only found in a single group of protozoa and then some animal cells. So some of the cells in our body, for example, would have cilia. So once again, these aren't on all eukaryotic cells, but some. The glycocalyx, this would be found on some eukaryotic cells, and it would be the outermost layer. So when you're looking at the cell, it's the outermost layer, and it would be the one that comes into direct contact with the environment. It's made up of polysaccharides, which hopefully you remember is a bunch of single sugars joined together, and they can appear in a number of different ways. They could be a network of fibers, a slime layer, or perhaps a capsule. And the glycocalyx, its function is to protect that cell, to help in adherence, so it could adhere to a surface or to another cell, and then signal reception, so it can actually receive a signal from outside the cell and send that into the interior portion. The next layer as we move in is another one that we would only see in some eukaryotes. I should go back. The glycocalyx you do not see in all eukaryotic cells, but you see in some. The cell wall is similar to that. Not all eukaryotic cells have cell walls. For example, protozoa and helminths do not have cell walls. But when we look at fungi, these do have cell walls, and they, just like we saw with the prokaryotic cells, they provide a rigidity and some structural support and shape. However, they differ in their chemical composition from those prokaryotic cell walls. Hopefully you remember that it had the peptidoglycan layer. In this case, you'd see thick layers of polysaccharide, sugar fibers that are composed of chitin or cellulose. And then you might also see this thin outer layer of mixed glycans. We have a picture of that. So if we're looking at the top, this would be the inside of the cell. The bottom here would be the outside, or outside to the environment. We start with the cell membrane. We then have this cell wall, which would be made up of the chitin, glycoprotein, which is sugar and protein bound together, mixed glycans, and then last of all is that outside glycocalyx that it may have. Okay? And then as we move inwards, we're going to start seeing some of the organelles or parts to a eukaryotic cell that all eukaryotic cells have. And obviously the cytoplasmic membrane, because it is always found in any cell, it would be found in the eukaryotic cell. It's just like the phospholipid bilayer that we looked at in the prokaryotic cell. In addition, it will have proteins that can be embedded in the layers, and these will often be transport channels or receptors. It also will contain sterols, which can help in rigidity and stability of the membrane, and this is really important for those cells that don't have the cell wall. And then lastly, it obviously serves as a selectively permeable barrier, letting some things in and keeping other things out, and vice versa. I'm going to stop here and post this as the first of our 2 mini lectures of what would have been my Monday lecture. And I'll finish up with my second one, with the second one going through the organelles. Okay? If you have any questions, please let me know.